Hello everyone, I am Ritik Naran Mahana, MBBS student at MKCG Barampur. You are watching MBBS VidNotes. Let's start.
Vitamin A is a fat-soluble vitamin. It has two forms, preformed form and provitamin form. Preformed form is the actual vitamin A, and the provitamin needed to be converted to actual vitamin A through some processing. Preformed are retinoids which have come from animal sources. It has beta-ion ring and isoprenoid side chain. In provitamin, it is called carotenoids. It comes from the plant sources. Here is the structure of beta carotene. The beta carotene, if we click from here, there is a formation of two retinoids here. But this is theoretically. In biological system, there is only formation of one retinoids. There are different types of retinoids like retinol, retinoic acid, and retinol. The retinol are of three types: all trans retinol, which is called also known as vitamin A1, 11-cis retinol, which is present in rhodopsin, vitamin A2, which is found in the fish oil, and here extra double bond is present in the ring. In our book, there is mention of three types of retinoic acid. One is all trans retinoic acid, 9-cis retinoic acid, and 13-cis retinoic acid. All these are used in more growth and morphological modifications and cell growth, and this all act like a hormone. Retinoic acid primarily act like a hormone, and retinol are for reproduction. The carotenoids are mainly primarily called beta carotene. Not all carotenoids are precursor to vitamin A. Some carotenoids are there which doesn't make vitamin A, like lutein and zeaxanthin, which is used as a medicine of macular degeneration, and lycopene, which is used in prostate cancer. Here we can see the relation say between retinol, retinal, and retinoic acid. The retinol is reversibly oxidized using retinal reductase to retinal. The beta carotene using dioxygenase in the intestine converted into retinal. You can remember beta carotene from carrot, useful for eyes. Eyes means aankh means A. That means which retinal, you can use any type of name. That means in from plant source we get retinal. Further we will see the metabolism inside the intestine. But remember, the beta carotene produces retinal. Then it is irreversibly oxidized to retinoic acid. From plant that we eat, we get beta carotene. Beta carotene in the intestine is digested by dioxygenase to give retinal. The retinal is then converted into retinol. And from animals, there are already preformed retinols in the form of retinol esters. So there also comes a retinol. Complete retinol pool is esterified by fatty acid to make retinol esters. The retinol esters is incorporated into chylomicron. The retinol chylomicron complex is then absorbed by the intestine. The retinol, which is incorporated into chylomicron, reaches to liver, where the liver cell uptake the retinol via ApoE receptors, where in ApoE is the ligand in this receptor. Once uptaken, it is stored as retinal esters, generally retinal palmitate. The cell in which they are stored is called hepatic stellate cell, which is present in perisinusoidal spaces. So they are also called as perisinusoidal cells. When there is a need of vitamin D, it is uptaken from the liver as retinol. The retinal palmitate is converted into retinol, and it is incorporated by two proteins, carrier proteins. One is retinol binding protein and another is transthyretin. So it makes a trimolecular complex. In this trimolecular complex then reaches to eye, skin, and other organs in which they are required. In eyes, we have to learn about the world visual cycle. The eye, there is we know there is presence of rods and cones. In rods, there is presence of rhodopsin. The rhodopsin is made up of 11 cis retinal and opsin. The rhodopsin, when the light strikes, is converted into all trans retinal. Going through all these steps, 11 trans retinal should be regenerated from all trans retinal. There are two ways that can be regenerated. From retinal epithelium, where transretinal is converted to cisretinal through retinal isomerase, and other one is in liver, where transretinol is converted into cisretinol. This is retinol, this is retinal. By retinol isomerase. So here transretinal needed to be converted to transretinol, which is done by alcohol dehydrogenase, which is a zinc dependent enzyme. So the transretinol is then converted into cis-retinol, then again to cis-retinal, and transported to the blood to cis-retinal, and 11 cis-retinal is formed. So the photoreceptor cells get their supply of 11-cis retinol from retinal epithelium. So, but actually, all this conversion of 11-cis retinol to all transretinal means during all this conversion, there is an important step called meta-rhodopsin 2. The meta-rhodopsin 2 is useful to activate transduction protein. The transduction protein, when activated, activates phosphodiesterase from inactive state to active state, which in turn converts cGMP to a 5' GMP, which closes the sodium channel. When the sodium channel get closes, it causes hyperpolarization. Hyperpolarization uh, polarization is on other places. The depolarization causes action potential generation, but in eye, the hyperpolarization causes action potential generation. Here is the same thing. The light strikes the rhodopsin. The rhodopsin get activated. It is the transduction. The transduction alpha, beta, gamma subunits. Only the alpha subunits got bind to phosphodiesterase, and phosphodiesterase converts cGMP to GMP. Work of cGMP is to open the sodium channel. When the cGMP level is lowered, it is closed. So it causes hyperpolarization and in turn action potential generation. The red glasses is used in fluoroscopic X-ray to protect the eyes of the doctor. The fluoroscopy X-ray is the technique in which there is a video of X-ray is drawn out, not a photo. It is required when we are doing live surgery or when you are doing something which requires the motion of the joints. We need to know how the action potential should be stopped. To do this, the activated rhodopsin should be stopped from binding to transduction, which is done by rhodopsin kinase enzyme, which actually phosphorylates serine residue of the rhodopsin, by which the beta-arrestin molecule binds to it to inhibit it. The cones are made up of conopsin. The conopsin are of three types: cyanoconopsin, iodoconopsin, and porphyroconopsin, which is responsible for the three colors: red, green, blue, RGB. Now let's know about gene regulation by vitamin A. We know retinoic acid acts like a steroid hormone. There are two types of retinoic acid: 9-cis retinoic acid and all-trans retinoic acid. The all-trans retinoic acid binds to retinoic acid receptor, which is a nuclear receptor, and then it binds to the response element that is present in the DNA. But 9-cis retinoic acid binds to retinoic X receptor, which is a nuclear receptor. Then it also binds to another nuclear receptor that is known as vitamin D receptor and make dimers. And then this dimer binds with response element of the DNA. So the vitamin D is required to the action of the 9-cis retinoic acid. Now let's know about the function of vitamin A. It is required for the maintenance of normal epithelium and normal reproduction. Because of maintenance of normal reproductive epithelium, it is antioxidant property. Generally, actually, beta carotene. The alteration of gene expression by retinoic acid, already known steroidal action. The embryogenesis. In embryogenesis, the differentiation of the three germ layer is required vitamin A. Vitamin A has immunological function also. The T cell is converted into regulatory T cells by retinoic acid and TGF. It is done by dendritic cells in the gastrointestinal tract. Deficiency of vitamin A causes decrease in the hematopoietic stem cells. So, except dietary deficiency, what are the conditions that can lead to the vitamin A deficiency? Like obstructive jaundice, liver problem by which the retinol binding protein decreases, and chronic nephrosis. The deficiency manifestation in the eyes are: the earliest manifestation is the loss of the green light sensitivity, then it starts night blindness, nyctalopia, then xerophthalmia. Xerophthalmia, we know about xerophytic gland, so dryness, actually dryness of the eyes. So the first dryness causes in the conjunctiva, then it starts go to the cornea. At the time the biot spots starts to form, it is a triangular, foamy, and present to the temporal region. For long term xerophthalmia causes keratomalacia, the softening of cornea. Here we can see the cornea is softened and dried. In skin also same, it causes dryness and keratinization. The follicular keratosis, phrenoderma or toad skin. It is caused by the keratinization of the epithelium, by which the plugging of adnexal gland occurs. The plugging of adnexal gland causes this called phrenoderma or toad skin. There is also keratinization and dryness to the mucosal secreting epithelium called squamous metaplasia. It happens in gastrointestinal tract, respiratory tract, but when it happens in the renal tract, there is a high chance of formation of renal calculi. We knew about the deficiency. Now let's talk about the toxicity. Generally, the Arctic explorer are susceptible to vitamin A toxicity because they eat the liver of polar bear, which is very much rich in vitamin A. The organ that is affected is lysosome, which in acute condition it causes pseudomotor cerebri. That means increase in the intracranial pressure, and hepatomegaly. The in chronic condition it causes headache, dizziness, anorexia, and bony exostosis. It causes very painful bone swelling. In toxic quantity, vitamin A can be a teratogen to pregnant mother. Generally, 13-cis retinoic acid is the teratogen. The vitamin A can increase the risk of bone fracture because it increases osteoclastic activity. There is a difference in hyperkeratonemia and jaundice. In jaundice, both skin and sclera becomes yellow, but in hyperkeratonemia, only skin becomes yellow. These are RDS of man, child, pregnant mother, and in lactation. How we can assess the vitamin A deficiency? It can be done by dark adaptation time, which is increased, the retinol binding protein quantity decreased, and also we can do some serum assessment. The serum assessment are of two types. One is a car and price reaction, in which the retinoids are bound to antimony, which gives a blue color. And other one is the spectrophotometry. The richest source of vitamin A is the halibut liver oil, and the second richest is cod liver oil. So this is all about vitamin A. Thank you for watching MBBS VidNotes.